Yeah, this is the third class for the GI tract and the last class for the digestive system. We did go through the anatomy and then secretions. Today we are going to study the digestion and absorption in the GI tract. So we'll be studying a little bit about anatomical basis for gastrointestinal absorption and the major mechanisms that are responsible for digestion and absorption of carbohydrates, proteins and fats. So the food is majorly composed of these compositions. One is carbohydrate, protein and then fats. Carbohydrates are polymers of sugars, right? Saccharides. Whereas fats are made up of glycerol and fatty acids, and proteins are polymers of amino acids. So when the digestion occurs to break down these big macromolecules into their monomeric or their building blocks. And most of it, the digestion occurs by hydrolysis. That means the water is involved in breaking down all these food particles. So hydrolysis of carbohydrates. In that, carbohydrates are present as polysaccharides or disaccharides, and there are specific enzymes in the digestive juices that hydrolyzes polysaccharides and disaccharides into monosaccharides. And mostly this is the base formula through which the digestion occurs by hydrolysis. The bond between the two monomers is broken down by H2O, and you get ROH and RH. That means the H2O is broken down into hydrogen ion and hydroxyl ion, and each monomeric part takes one of the ion. Hydrolysis of fats. Fats in the diet are usually triglycerides, that they are mostly neutral fats, and fat digestive enzymes fix the fat into fatty acid and glycerol molecules by hydrolysis. Again, proteins are usually made up of amino acids, so they are mostly a long polypeptide chains because the bond between the amino acids is peptide bond. So those peptide bonds are broken down and you mostly get amino acid. The final product is the amino acids, but the breakdown occurs like it can be broken down into dipeptide and tripeptide, and then finally broken down into amino acids. And same mechanism is hydrolysis here. So starting with the digestion of carbohydrates, the diet carbohydrates in human diet, most of the carbohydrates which are present are in the form of either sucrose, lactose or starches. Sucrose, like for example cane sugar, lactose which is found in the milk, and most of the non-animal foods are starch composed, like potatoes, grains. And other ingredients which are to the lesser extent are amylose, glycogen, alcohol, lactic acid, pyruvic acid, pectin, dextrins, and some other meat carbohydrates. And cellulose is other carbohydrate which we cannot digest. So where does the digestion of the carbohydrate start? It starts from the mouth. As I said yesterday, like the main secretion in the mouth is saliva, which has digestive enzyme ptyalin or amylase, and that's where the carbohydrate digestion starts. But only 5% of the starch gets digested in the mouth, mostly the starch to maltose. And then as the food along mixed with the saliva goes down from the esophagus to the stomach, it takes along with this a little bit of the ptyalin along with it. And then in the stomach, the starch digestion continues in the stomach before the food is mixed with the stomach secretion. So in the stomach, mostly the enzyme that is secreted is pepsin or pepsin, and that pepsin is active or can function only in the acidic environment. So HCl is released. So once the HCl is released, the ptyalin or the amylase becomes inactive. because amylase cannot function at acidic pH. So before the secretion of the stomach starts, the almost 30 to 40% of the starch gets hydrolyzed by the salivary amylase in the stomach. So the amylase which is secreted in the saliva, only 5% of the carbohydrate gets digested in the mouth, but the rest of the starch, almost 30 to 40%, is digested in the stomach with the help of the same salivary amylase. Because once the acid is secreted, the amylase becomes inactive, so it has to be before the acid or the stomach secretions, gastric secretions starts. Now going to the pancreatic, going to the small intestine. So in the small intestine, as I said, it is a sphincter of Oddi which allows the secretions from the common bile duct and the pancreatic duct, which combine together to form capilla of Vater's, and they do their secretions through sphincter of Oddi in the duodenum. So that's where the pancreatic juices are secreted, and that pancreatic juice has pancreatic amylase, which is again a carbohydrate digesting enzyme. In duodenum, it hydrolyzes the remaining carbohydrates in the chyme to maltose and all other small glucose polymers. Then, as it moves down, villi enterocytes. In the villi, there are small cells which are called the epithelial cells. These are called enterocytes, which line the villi. Those contain 4 different enzymes: lactase, sucrase, maltase, and alpha dextrinase. You see this, you can say that these are all disaccharides, right? So lactose is a disaccharide, sucrose, maltose, all acts on a disaccharide molecule. So until the pancreatic amylase, all those things are broken down mostly into disaccharides. And at the villi, in the enterocytes, they have particular specific enzymes which can break down those disaccharides into monosaccharides. And those monosaccharides are what? Are absorbed into the cells in the small intestine. So a brief summary here. Starches by thiolin, which is produced by saliva, 20 to 40%, 5% is in the mouth, and rest 30 to 40% is in the stomach. And then pancreatic amylase, which is almost 50 to 80% of the starch is digested by pancreatic amylase in the small intestine, duodenum mainly. And all those are mostly digested, all the starches are digested to disaccharide forms. So maltose and 3 to 9 glucose polymers. All these are again digested at the villi by a specific enzymes, disaccharidase enzymes, into glucose. And this glucose is and galactose, which is converted to glucose, or fructose, which is again converted to glucose. So mostly the glucose, you know, the glucose is the main molecule for the energy formation, right? So most of it comes back to the glucose, which is used for the energy. So glucose is the molecule that enters into the that is absorbed by the small intestine. So digestion of the proteins. Dietary proteins are usually long chains of amino acids which are bound together by peptide linkages. So this is a peptide linkage between CO and NH group. This is the peptide linkage here. And upon hydrolysis, when you add water, they are broken down such that hydrogen ion is bonded with one of the amino acid, whereas hydroxyl ion is bonded with one of the amino acids, such that it forms an amino acid at both ends. So hydrogen is bound to the amino, and whereas OH is bound to the carboxyl. So protein characteristics is dependent on the type of the amino acid. You know whether the protein is hydrophobic or hydrophilic. It's based mainly on the type of the amino acid it is composed of. In the sequence, what type of amino acids are there? What is the composition of hydrophobic amino acids or neutral amino acids or hydrophilic amino acids? Based on that, the protein type or the protein characteristics digestion of protein starts in the stomach and then extends into the small intestine. In the stomach, mostly pepsin. Pepsin, which is formed as pepsinogen. Pepsinogen is an inactive form of the pepsin, and this is activated by acidic pH. The important feature is the digestion of the collagen, major constituent of the connective tissue in the meat. So majorly what it does is it breaks down the collagen such so that collagen is the ECM material, mostly the extracellular matrix which holds the cells together to form the tissue. So that is broken down, so it is easy to break down the other food part. So important to digest collagen for other enzymes to penetrate, and digest initiates the process of protein digestion. So 10 to 20% of the total protein is digested to proteases, peptones and few polypeptides. So mostly almost 10 to 20% of protein is digested in the stomach by stomach pepsin. Rest is digested in the small intestine, where the pancreatic secretion are released, and the pancreatic secretion are released, as I said, again in the duodenum at the sphincter of Oddi, and those pancreatic secretions have the protein digesting enzymes, mostly trypsin, chymotrypsin, carboxy, polypeptidase and proelastase. So trypsin and chymotrypsin split molecule into small polypeptide. Carboxy polypeptidase, as the name indicates, it works from the carboxyl end of the polypeptide chain. So it starts removing the amino acid from the carboxyl end, and then proelastase is converted to elastase, which digests the elastin fiber. Elastin fiber is again a fiber as a ECM material that holds the cells together. So if you have a meat type of material, collagen and the elastin are the major components that has to be digested before any enzyme can penetrate into digestive. So most products of protein digest are dipeptides or tripeptides with few digested to amino acids. So they are all dipeptides or tripeptides. So before they have to be absorbed, they have to be converted to amino acid. So at the Bruch borders, that means where the villi are there, as I said, at the villi you have a lining of the enterocyte cells, and the enterocyte cells are where the final digestion into the monomeric or building block happen, and that's where the absorption also occurs. So at that place, the peptidases in the enterocytes helps in digesting the final products of the protein digestion. And two types of peptidase enzymes are there: amino polypeptidase and dipeptidases. So dipeptidases break down all the dipeptides, whereas amino polypeptidase that would break down the polypeptides from the amino end. So each amino acid would be removed from the amino end. So it splits all the large polypeptides into tripeptide and dipeptides and few into amino acids. So amino acids plus dipeptide and tripeptides are easily transported through the microvillar membrane to the interior of the enterocytes. So they can enter into the enterocytes at the Bruch border, that is at the villi. The cytosol of the enterocytes has some peptidases that can break down the dipeptides and tripeptides into their constituent amino acids. So proteins are broken down in the stomach by gastric pepsin into proteases, peptones and polypeptides, and these are again broken down by trypsin, chymotrypsin, carboxy polypeptidase and proelastase into polypeptides and amino acids in the duodenum, that is in the small intestine. And then in the small intestine, in the enterocyte, the dipeptides, tripeptides and some of the amino acids can enter into the enterocytes. In the enterocytes, you will have all the small peptidases which can break down these dipeptide and tripeptide into further down into the amino acids. So the final product of amino acids is what you will get. Digestion of the fat in human, as I said, most of the fats are neutral fats known as triglycerides. Triglycerides means it has 3 chains of fatty acid which is glycerol molecule. and most of them are phospholipids, cholesterol esterols, and cholesterol sterol compounds. These are the three major compounds. So there is a small lipase or a minute amount of lipase which is also secreted in the lingual, that means at the tongue, that is secreted, produced by lingual glands of the mouth and is swallowed with the saliva, which is very less than 10%. And emulsification initiation occurs, that is breakdown of the fat globules into the small cells. As I said, fats are not soluble in water, and the enzymes that have to act on the fats are water soluble enzymes. So it is necessary that the fats have to be made to be water soluble. And how that is made to be water soluble is where the bile comes to help. Bile has the ions that comes to help to make them water soluble, so that the water soluble enzymes can come, and enzyme that is mainly the lipase can come and act on those fat particles and break down. So mostly that occurs in the small intestine. In the small intestine, where at the sphincter of the OD, when the pancreatic secretions occur, before the pancreatic secretion, as the food goes into from the stomach to the duodenum, the contraction of the gallbladder occurs by the control of the hormone, and as the gallbladder contracts, the bile is secreted from the common bile duct into the duodenum. So when the bile is secreted, that bile has the particles which are amphoterics, that means they have hydrophilic end and hydrophobic end. So the hydrophobic end binds to the fat, and hydrophilic end is directed towards the outside, to the aqueous environment. So that way they bind to the fat, all the fat molecules they will bind and break down into the smaller fat molecules, so that it's easy for the enzyme to digest them. So big fat globules are divided, or they are partitioned into small fat globules along with these bile salt molecules, which help them to be more water soluble, and those small particles are called micelles or micelles, M-I-C-E-L-L-E. It will come. So that is, and the process is called emulsification. Emulsification for water soluble enzymes to act on them. Bile has large quantities of bile salt and lecithin. A phospholipid, polar portion of the bile salt and lecithin are highly soluble in water, while the remaining portion is fat soluble. So fat soluble portion of the bile secretion dissolves in fat globules, while exposing their water soluble regions, making the fat globules soluble as well. So the triglycerides are broken down by pancreatic lipase and enteric lipase. Pancreatic lipase produced in pancreatic secretions, and enteric lipase is which is produced in the enterocytes, that is at the small intestine. This is the pancreatic secretion is at the early part of the small intestine, that is duodenum, and this is the enterocytes, that is where mostly the absorption starts. So this is a glycerol which is connected to the 3 fatty acids. Upon when the lipase acts, it breaks down into diglyceride with removing 1 fatty acid, and further it breaks down into monoglycerides and fatty acids. So the final product of fat digestion is mostly monoglyceride and fatty acid. That means glycerol molecule binding to 1 fatty acid and 2 fatty acids are removed. So digestion of the cholesterol ester and phospholipids. Nice cholesterol is present as cholesterol ester, that is cholesterol plus fatty acid, and phospholipids is phosphorus atom plus fatty acid and glycerol. Specific enzymes in pancreatic secretion and cholesterol esterase hydrolase, which hydrolyzes cholesterol ester, and phospholipase A2, which hydrolyzes the phospholipids. So these are the 2 other forms of the fat that are digested in the small intestine. So this is the main process. We have big fat globule. These are the bile salt that has hydrophobic and hydrophilic ends. So these bile salts surrounds this fat globule and divides the fat globule into small molecules, small droplets, and binds in such a way that the hydrophilic ends are exposed to the aqueous environment, where the hydrophobic ends are binding to the fat molecule. And this is how they can easily allow the lipase to enter and start digesting the fat molecule. So this is the same thing. You have fat and the bile salt emulsification, emulsified droplets here, and then these are digested by the lipases. And when the lipase digests, it digests into the free fatty acids and monoglycerides. And these free fatty acids and monoglycerides can easily go inside the enterocytes, and once they go inside the enterocyte, they mostly enter the endoplasmic reticulum. In the endoplasmic reticulum, they are both combined again to make triglycerides, and those triglycerides are then released from the endoplasmic reticulum with the help of a protein that surrounds those triglycerides, forming the chylomicrons. These are the chylomicrons. So this is mostly the protein and the triglyceride. And these triglycerides are the ones which are formed by the monoglycerides and the fatty acids that enter the endoplasmic reticulum. You know that some of the endoplasmic reticulum includes, that is packaging of the proteins and all those things, vesicle formation and everything, right, from the endoplasmic reticulum body. So that's what happens, and that gets packaged into chylomicrons, and these chylomicrons are, when they on the other end, this is the lumen side of the intestine, and this is the basal side of the intestine, where the lymphatic vessels and the blood vessels are there. So most of the fats go into the lymphatic vessel. So from the inside of the enterocyte, it is released through the membrane into the at the basal side, and they migrate into the lacteal. Lacteal is the major lymphatic vessel in the intestine, in the villi mostly. And from there it goes into the hepatic vessel to go through the blood vessel. So to go into the blood circulation, the fat doesn't directly go into the blood circulation. Glucose and amino acid directly go into the blood circulation, but this goes to the lymphatic system, and then from the lymphatic system through the hepatic duct it goes into the thoracic duct or hepatic duct. Then it goes into the blood supply. So this is what I had told yesterday about the foldings in the small intestine to increase the surface area. Okay, it's the same thing. Here you have the folding, circular folds. First is the circular folds, which is called valvulae coniventes, or folds of Kerckring. And then you have those folds again are folded into villi, and each villi is again having the microvilli on its end. And these microvilli are the extensions of the enterocytes. So this majorly increases the absorptive area to about 1000 folds, and it can be equivalent to size of the tennis court. And it begins in the mid-duodenum, extends all the way to jejunum, and eventually disappears before it reaches the distal ileum. So in the colon you won't see all these villi or microvilli. It's mostly only in the mid-duodenum, then jejunum, and then a part of the ileum. But as the ileum is close to the colon, you don't see those villi and microvilli. So in the villi, if you see the structure, this is how the circulation. We already have seen this in the first class, I think. You will see these are the veins and the arteries with network here, and in the middle you will see a lacteal, that is a central, that is one of the lymphatic vessels. So these are the blood capillaries, and you have the lymphatic vessel to which the fats are transported. So apart from carbohydrate, absorption of their products, water and ions are also absorbed. Water is, as I said, like most of the water which is secreted or which we drink is helpful, it is secreted, it helps in digestion, but again it is absorbed back into the system to maintain the hydration of our body. So but it obeys the principle of osmosis. So osmosis is wherever the concentration of your ions are more, the water starts moving there. So yesterday I said that the water mostly follows the sodium ions, right? So it would start following the sodium ion. So sodium ion, when it goes in negatively, it also pulls a bit of sodium is positive, so it also pulls chloride ions along with it. So it forms a sodium chloride ions. So you have an ionic concentration gradient formed, and because of that ionic concentration gradient, water also starts moving along with sodium. So when chyme is diluted, water is absorbed through intestinal mucosa into blood vessels of the villi. When hyperosmotic solution comes from the stomach into the duodenum, water is transferred from the plasma to make chyme isoosmotic with the plasma. So what way the water should move depends on the concentration of the chyme. So if the chyme is dilute, then the water is absorbed. If the chyme is hyperosmotic, that means less water is there, then the water is secreted into the wherever the chyme is, so that it can be more isotonic to the plasma. And if you see, these are the enterocytes here. This is called paracellular spaces here. This paracellular spaces is where the sodium starts moving out. And as the sodium starts moving out, along with it, it also takes a little bit of chloride ions. Because of the positive charge, it drags the chloride ions. And this sodium transport is an active transport of sodium from the interior of the cell to the paracellular spaces on the side as well as on the back. So as they move, the concentration of sodium increases outside. So the water is dragged from the lumen into the cell, and from the cell into the inside of the small intestine. So it follows the route of the sodium transporter. So sodium moves out, along with comes the water. And the aldosterone is the hormone which is produced by adrenal gland cortex, that also enhances the sodium absorption. When you are dehydrated, aldosterone is secreted, and aldosterone activates the sodium absorption. So when the sodium absorption is activated, the water is absorbed more. So bicarbonates, yesterday I said, like when the ducts, when the pancreatic secretions from the acids start moving into the pancreatic duct, at that time you will see a lot of bicarbonate ions released into those pancreatic juices along with the along the duct. So at the same way, when the bicarbonates, there are a lot of bicarbonates in the juices in the secretion. So those have to be absorbed back. So pancreatic secretion and bile secretion both contain high amount of bicarbonate. So that has to be absorbed. The mechanism is very indirect, that is active absorption. When sodium is absorbed, in exchange, hydrogen ions are secreted into the lumen, which combine with the bicarbonate in the lumen, which combines with the bicarbonate and forms a carbonic acid. And this carbonic acid now dissociates into water and carbon dioxide. And that water mixes with the chyme, whereas the carbon dioxide is absorbed by the blood and then it is exhaled through the lungs. So when the sodium is being absorbed into the cell, at the same time, because of the positive ion being absorbed, the positive ion is secreted out. So it absorbs in the exchange of the hydrogen ion. So hydrogen ion is released into the lumen, and that hydrogen ion combines with bicarbonate and forms carbonic acid. And that carbonic acid is broken down into water and carbon dioxide. The water is mixed with the chyme, whereas the carbon dioxide is absorbed by the blood supply and then it is released through the lungs. Also, in exchange of absorption of the chloride ions, bicarbonate is released in ileum and colon. This bicarbonate neutralizes the acid which is produced by the bacteria in the colon. So as I said, like in the first class, that there is some bacteria in our body, in the intestine, in the colon, which also helps to break down certain type of the food, and that can produce acid. So this bicarbonate which is released in the ileum and colon helps to neutralize that acidic effect which is produced by the products of the bacteria. Absorption of the other ions like calcium, iron, potassium and magnesium, phosphate and some other ions occurs in the same way. Calcium is actively absorbed into the blood from the duodenum, and it is regulated by parathyroid hormone and vitamin D. Parathyroid hormone activates vitamin D, which enhances the calcium absorption. Iron which is actively absorbed from the small intestine uses hemoglobin formation. So iron is needed for the hemoglobin. So that is also absorbed in the small intestine. All other ions are also actively absorbed through the intestinal mucosa. Do you understand active absorption? Active absorption is when there is a use of energy, right? So at all these active absorptions, there is use of energy. That means ATP is broken down at each point. And monovalent are most readily absorbed than the divalent molecules. So these are some of the, it's a table which describes of the absorption of the different molecules and what the location of the major absorption sites in the small intestine. Like this is sugars like glucose and galactose are absorbed in the upper and majorly in the mid small intestine and lower. So there is no absorption in the colon. So there is no absorption in most of the items in the colon except the sodium is absorbed in the colon because sodium absorption is necessary for the water to be absorbed. So major absorption of the water occurs in the colon. So mostly in the colon it's mostly only the sodium and a little bit of chloride ions that are absorbed. So these are just the summary of the digestion and absorption together. You see, the major sources of carbohydrates are sugars, sucrose, lactose, and starches, and glucose accounts for 80% of the carbohydrates. Salivary amylase and pancreatic amylase break down the starch, essentially converting all starch to maltose or short polymers of glucose before the time reaches the jejunum. And Bruch borders, that is the villi hydrolases like lactases, sucrases, maltase, and all these disaccharide digesting enzymes then converts all the carbohydrates to monosaccharides for absorption. This is just a summary here. So how does the absorption of the glucose occur? Glucose absorption is again dependent on sodium, or sodium is co-transport. Glucose acts as a co-transporter for the sodium. So for the sodium to enter, it needs to be needs to go through the transport molecule, transport protein. And for the transport protein to take the sodium has to be bound to some molecule, and that molecule is glucose. So sodium binds to glucose or galactose and then passes through the transport molecule and enters into the interior of the enterocyte. And from the interior of the enterocyte, it actively gets transported into the blood. So this is facilitated diffusion because it takes the help of the transport molecules. For the proteins, I said that protein digestion starts in the stomach by pepsin, and in the stomach pepsin cleaves the protein into smaller proteases, peptones or polypeptides, and gastric chyme, when released into the duodenum, it gets exposed to the pancreatic secretion, which is rich in proteases such as chymotrypsin, carboxyl peptidases, and elastase. So all these things again digest all the proteins, polypeptides into the di and tripeptides of amino acids. And most of these trypsin, chymotrypsin, carboxypeptidase, all these are released in an inactive form, and then they are activated. And most of the chymotrypsin, carboxypeptidases are activated by the help of trypsin. Trypsin is released as trypsinogen, which is broken down to form the trypsin, and trypsin is the active form. That trypsin helps to activate chymotrypsin carboxypeptidase. So once all those things break down the peptides into dipeptide, tripeptide, and amino acids, then the digestion of the protein is followed by the absorption of all these dipeptide, tripeptide at the enterocytes. And in the enterocytes, there are aminopeptidases and some dipeptidases and tripeptidases that break down all the remaining small polypeptides into monomeric amino acids. All these, as you said, like all the monomeric amino acids are formed in the enterocytes, inside the interior of the enterocytes, and from the interior of the enterocytes, they are again transferred to the blood supply through the basal site. These are, you don't have to know about this, but this is all the transporters that are used for the absorption of the proteins. So there are a lot of transporters on the absorptive side as well, on the absorptive side of the enterocytes as well as on the basal side of the enterocyte. So these transporters helps for the uptake of the molecules, protein molecules into the cell, and there are transporters at the basal level which helps the protein molecules to get into the blood supply. And there are some transporters which are also involved in reducing the absorption. When there is no need, it reduces the absorption of the protein, amino acids and dipeptides. So with the digestion of the lipids and absorption of the lipids, so fats are converted into emulsified fat, and emulsified fats are the ones that are acted upon by the enzyme lipase, and that breaks down into fatty acid and monoglycerides. Fatty acid and monoglycerides are taken into the enterocytes. In the endoplasmic reticulum, they are again combined together to form triglycerides, and then the type of protein that encloses the triglycerides forms a chylomicron, and that chylomicron is again absorbed at the basal region into the lymphatic system through the central lacteal, which is present there, and from the central lacteal it goes into the blood supply. This is the same thing. You see hydrophobic ends and hydrophilic ends shows here, and this is the same thing we discussed. These are cholesterol molecules and cholesterol esterol molecules. So major fatty acids are saturated, monosaturated, polyunsaturated fatty acids. Saturated fatty acid, lauric, myristic, palmitic, and stearic. I don't think you need to know all these details unless you have other class which talks most about the composition of the food. Different types. There are different types of fatty acids based on the like number of the hydrogen molecules that are present in the fatty acids. I think this is a major summary of all the digestions carbohydrate, protein, and fat digestion, and then where the site of the digestion occurs, and what is the part of the absorption. This is just the summary of what we went through today. So most of the food is absorbed in the small intestine. Colon is a place where it is mostly the water that is absorbed, and it is also helps in absorption of the electrolytes and the ions, and it also helps as a storage place. So most of the proximal colon, that is which is close to the small intestine, is an absorbing colon, whereas the distal colon acts as a storage colon. So absorption of the sodium and chloride ions create osmotic gradient for the absorption of the water that we already discussed. So tight junctions between the epithelial cells are tighter, thus they prevent the diffusion of the ions. Distal colon secretes bicarbonate ions in exchange of the chloride ion absorption, and bicarbonate helps to neutralize the acidic products that are formed from the bacteria. This also we read. So numerous bacteria are present, which are most common ones are colon bacilli, and these can digest cellulose, which we don't have any enzyme in the human body to digest the cellulose. So most of the cellulose is digested by the bacteria which is present in our colon, and it synthesizes, bacteria also synthesizes vitamin K, which helps in blood coagulation, and it also produces vitamin B12, thiamine, riboflavin, and various gases. So these are all the things that are the useful functions that the bacteria does for us in the colon. The brown color of the feces is stercobilin and urobilin, which are the derivatives of bilirubin. Bilirubin is a degradative product of the hemoglobin, as I said, and it is mostly yellow in color. The bilirubin is yellow in color, which is a degradative product of the hemoglobin, and the derivatives of bilirubin are stercobilin and urobilin, and that's the brown color of the feces is because of those two. And each person's colonic bacterial flora is different. It is based on the type of food you take usually. So if you're non-vegetarian or vegetarian, your the composition of the bacteria in your gut will be different. Yeah, it is early. I think this was a short one.